All right, we are going to talk about cell transport today. The focus is going to be on the cell membrane, but we are going to talk about we're going to first start off with the structure of the cell membrane, um, but then we're going to talk about specifically how things get in and out of the cell, because the cell membrane's main function is to be able to control what goes in and out. But it's not the only function of the cell membrane. You can see from this slide, it actually has other roles. Uh, but we are going to focus on the regulation of materials going in and out of the cell. So first off, what is the cell membrane? It's actually a bilayer. So the cell membrane defines the cell. It's made up of two parts, kind of like a sandwich, two pieces of bread. The individual component of the cell membrane is something called a phospholipid, and it's made up of two parts. There's a head and there's a tail. You can see the tail. The tail is hydrophobic, does not like water, like oil, for example, is hydrophobic. And the hydrophilic end is something that is actually attracted to water. So the way it's oriented is the hydrophilic end faces water. That would be basically the outside of the cell. So if this is a cell, then this would be outside, also called the extracellular fluid. And then this would be the inside, called the intracellular fluid. So the hydrophilic ends would face inside and outside, because that's where the water would be. And the hydrophobic tails would face towards each other, away from water. If you remember, like a vesicle is actually like a little sac that is used by the cell to transport things. It would actually just simply be the bilayer folded up into a little circle, and that would be used to transport things within the cell. So you can think of like a vesicle, like a little mini cell in a sense. Okay, now when you think about the cell membrane, it's not exactly a solid. It's not exactly a liquid. It's something in between. I like to think of it as like a jello. Kind of like soft, but not too soft. If it was a liquid, it would just fall apart. But if it was a solid, it would be too stiff and rigid. Nothing, would be able to. It wouldn't be able to move. Nothing would get really through it. So think of it like Jello. Okay, and you can see this is the inside of the cell membrane. Now, the cell membrane, the main component is the phospholipid, but there are very important proteins that are in the cell membrane that are going to have many roles. Again, the ones that we're going to focus on are in regards to transport. However, there are other roles that we will highlight. Here's a neat picture of the bilayer. Okay, you can see that bilayer beautifully here. This is the red blood cell. So this is the intracellular fluid that's inside. This would be the extracellular fluid, and you can see that bilayer of the red blood cell. So there's that terminology, intra versus extracellular fluid. So you can also think of the intracellular fluid like as the cytoplasm, for instance. You should be familiar with that term. Here's another picture of this. So you can see the cell membrane is predominantly made of phospholipid. Again, that's the thing that has a head and a tail. The tail is basically fat. Lipid is another word for fat. That is, again, hydrophobic. So it's facing inside, it's facing away from the water. The tails are facing towards each other, whereas the hydrophilic end is facing either outside or inside where there's a lot of water. But you can also see in this picture that it's not just the phospholipid bilayer. There are other things that are in the cell membrane that obviously have different roles, and we'll highlight again some of those roles. I do want to mention cholesterol because that does come up. In animal cells, cholesterol is actually a stabilizer. So it actually keeps the cell membrane like jello, not too hard, not too soft, not too much like a solid, not too much like a liquid. It keeps it in a good state. Um, Interestingly enough, plant cells don't have cholesterol. So if you think like plant oils, read the back of the label, they're cholesterol free. So they must do something different Because if cholesterol is the stabilizer for animal cells, then what do plant cells do? They must be doing something different. We won't touch upon that in grade 10, but if you're interested in this topic, it actually has to do with the way they regulate the types of fat in the cell membrane. Because I did say the tail is basically a fat. It's a lipid. It's called phospholipid, right? So the lipid is another word for fat. And there are different types of fats, and plant cells have the ability to alter the types of fat to keep the cell membrane like jello. But we're not going to talk about that in grade 10. Here's a nice picture of a cell. You can see this is like a little vesicle. So this would be made up of that bilayer. You can see that the cell membrane does not have a defined shape to it. You can see like it's not a perfectly smooth. You can actually see here it's actually folding in, probably to swallow up something. I have no idea what it's trying to swallow up. You see here it's reaching out a little bit. And again, it would not be able to do this if the cell membrane was like a solid. It wouldn't be able to do it. If it was a liquid, it would just fall apart. So it's got to be something, like we said, in between. Okay, talked about cholesterol. Now, I do want to highlight some of the other roles that the cell membrane has, because there are special proteins in that cell membrane. Again, we are going to focus on transport, but I do want to highlight a couple of other things. So for example, this is an image showing you transport, and we'll talk about this, not today actually. The next time we'll talk about how proteins are involved in transport. You can see here's an example of this picture is of some molecules that are traveling through this protein. What you actually see in this picture is interesting. I actually should highlight this. You'll notice that there's two color, so it's a purple sphere and a pink sphere. So this is actually showing you that the cell membrane is very selective as to what can go through the cell membrane, because the proteins are specialized for particular things. You can see here that the pink spheres can go through, but not the purple ones. So there's obviously some sort of a screening process going on here. Here's another image of some sort of molecules traveling through the cell membrane through these proteins. You can see in this case, potassium is going one way and sodium is going the other way. We'll come back to this one later on. But just to highlight, so I did mention that proteins are also involved in other things other than transport. But once again, you see that here. So here's transport. This is actually how glucose gets into the cell. These are called glute transporters. So kind of really no different than what you're seeing here. But the cell membrane also contains proteins that act as receptors. And this is actually for communication. It's one of the other roles that the cell membrane is involved in, which I believe actually came up somewhere here. Yes, transmitting signals. So that's another way of saying communicating. So in going back to this slide, a receptor is basically a special protein that doesn't have to sit on the cell membrane, but in this case, it is sitting in the cell membrane. And what receptors do is they pick up other chemicals that are basically ways of communicating. So for instance, insulin is actually a chemical message, and the insulin receptor is what actually allows that chemical message to be received by the cell. So without that insulin receptor, this cell would not be able to receive that message. So receptors, in other words, are involved in communication. When cells receive chemicals from other cells, they need these special receptors to pick up these signals. So again, we are going to focus on transport, but keep in mind that the cell membrane does play a role and other things such as communication. Here, it's a protein that's involved in recognition. So another role of the cell membrane is in being able to, or allowing the immune system to recognize whether or not cells belong there. So the cell membrane does have molecules on the surface that actually allow the immune system to be able to identify those cells as whether or not they're foreign, which means they should be eliminated, or they are not foreign, that they belong there. Kind of like, I guess the analogy would be like a school uniform, right? So the school uniform's on the surface of the student, and on the surface of cells are special proteins that allow other cells to recognize whether or not they belong there. The cell membrane also has special proteins for anchoring. You can see that here, and you can also see that here. So why anchoring? Well, because we know that cells make up body parts like organs. And if cells could not anchor to each other, then what would happen is they would break apart. And that wouldn't be good for organs. Imagine organs, if the cells could not anchor to each other, they would just simply break apart, and that would be disastrous. So that's another role, right? Cells being able to anchor to other things, and that's obviously anchoring through the cell membrane. But again, the focus is going to be on transport. So it's said that the cell membrane is involved in controlling what goes in and out, and that's mostly true. It does not have control over certain things, though. Certain things are just going to be able to move in and out of the cell membrane. So some things like gases, water, things made of fat, that's what this means basically, is it's hydrophobic, doesn't like water. So other things that don't like water, like lipids, will be able to travel through the cell membrane. So these things can go right through the cell membrane. So there's a term here, it says selectively permeable. So what does that mean? Well, in some regards, the cell membrane is just simply permeable to things. What does permeable mean? It means it can go through. So if something is permeable, that means it can go through the cell membrane, whether that's in or out, okay? So gases can go in and out, water can go in and out, hydrophobic things can go in and out, all right? When you hear the word selectively permeable, that means, I guess you can think of it as not everything's going to be able to go in and out. So the things that cannot just go in and out, well, they're going to need special doorways. And that actually goes back to a picture like actually like this, or like this, or like this. These are, you know, this, and this. These are like special doorways that allow the cell to then control. So it does have some control as to what can go in and out. Some things it can't control, some things it can. So what we're going to do today, I'm going to wrap up. We're actually going to talk about the things that can just easily go through the cell membrane. And we're going to talk about two really important concepts when it comes to things that can move through the cell membrane. Really important concepts. And that is diffusion and osmosis. Next time, we'll talk about the use of proteins. And that's when the cell membrane actually has some control as to, you know, what it allows in or what it gets rid of. But today we're going to talk about osmosis and diffusion. So what is osmosis and diffusion? Well, first off, they are passive types of transport, which means it doesn't require any energy. Next time we'll see examples of active transport, which means in that case, energy is required. So we can actually classify transport as passive or active. Diffusion and osmosis are passive forms of transport that do not require any energy. So what are they? What is diffusion? Diffusion is just simply a natural process when a molecule will, or molecules that are in one area will spread out over time. So if I look at this beaker, you'll notice there's a high concentration of a particular molecule here. And as time goes by, you notice that the molecules spread out. This is diffusion. Diffusion is when molecules move from a high concentration to a low concentration. Well, my writing's terrible there. So that's just a natural process. No energy is required. It just happens on its own. Now, this is actually important because when a cell, for example, if you remember from, the parts of the cell, cells like a eukaryotic cell would have something like a mitochondria. Okay, remember the mitochondria is the organelle that makes energy. Now, what the mitochondria uses up is oxygen, and it produces CO2. So what that means is that inside a cell, eventually, there will be very little oxygen, but there will be a lot of carbon dioxide. And again, that's because the mitochondria uses up oxygen and doesn't produce, and it produces CO2. And whenever a trivia question, which cell doesn't have mitochondria, that would be the red blood cells because they want to transport the oxygen. But in any case, so if a cell has mitochondria, it uses up oxygen, produces CO2. So how does it get more oxygen in? Well, that cell would have to be exposed to a high level of oxygen. So when you breathe in, you're breathing in lots of oxygen. So what happens is the oxygen will diffuse into the cell. Now, why does it diffuse into the cell? Because when you breathe in, you're bringing in lots of oxygen, and the cell has very little oxygen, so it will just naturally diffuse into the cell. What's also true is when you breathe out, you're also breathing out lots of carbon dioxide. Why is that? Because the cell has lots of carbon dioxide, but the air that you're breathing in and out does not. So then the carbon dioxide actually will leave. And that's just by simple diffusion. So that's an important concept, diffusion. The ability of things to just simply move from a high concentration to a low concentration. So things that can move in and out of the cell freely, like gases, will just simply diffuse from a high concentration to a low concentration. Now, osmosis is specifically the diffusion of water, but it's not just affected by concentration. In other words, just moving from a high concentration to low concentration is a factor, but it's not the only factor. It will also move to an area of lower pressure. So you can literally force water by using pressure. And it will also move to the area of where there's more solutes. And that's actually what we're going to focus on. So what does that mean? More solute. So there are three important terms here that have to do with solute concentration: isotonic, hypertonic, hypotonic. What do they mean? So an isotonic environment is when, let's imagine you have a beaker and there's a cell in the beaker, and the solution that the cell is in, let's say the salt concentration is 1%, and the salt concentration, or I should say solute concentration, in the cell is also 1%. You notice that they're exactly the same. When the inside and outside concentrations are the same, that we say that the environment is isotonic, okay? The extracellular fluid outside the cell is isotonic to the intracellular fluid. If the outside environment has a higher solute concentration, then the extracellular fluid is hypertonic. If the inside environment has a higher concentration than the outside, then the outside is hypotonic. So hypotonic extracellular fluid means that there's less solute outside than inside the cell. It's the exact opposite of hypertonic. Now, what does this have to do with anything? Well, one of the rules is water will just simply go to the higher solute concentration. So if we have a beaker and the cell is sitting in the beaker and the solute concentration in the cell is, let's say, 2%, and outside the cell is 8%. This is assuming that it's the same type of solute, by the way, because it does get a little bit more complicated than this, but just to keep it simple, then water in this case will leave the cell because the outside of the cell has a higher solute concentration, and water moves towards the higher solute concentration or to the hypertonic side. So this is a normal red blood cell. This is what it normally looks like. When it's in an isotonic environment, that means that this is its shape, okay? Water will flow in and flow out at an equal rate. So it won't gain water, it won't lose water. However, if it's in a hypertonic environment, that means the outside has a higher solute concentration. So water will leave the cell and the cell will shrivel up. And the reverse is true if we put it in a hypotonic environment. But if it's in a hypotonic environment, that means the outside has less solute concentration. So the water rushes in and causes the cell to swell up. This is just images of plant cells doing this. The difference between plant cells and animal cells is that because a plant has actually a cell wall, it doesn't run the risk of actually bursting. Like this red blood cell can actually burst, whereas plant cells can't because the cell wall. So if we do have, for example, in this slide here, a cell that's accumulating lots of water, it doesn't run the risk of bursting because of that cell wall. We can actually pinpoint the isotonic value. So this is actually a graph of pieces of potatoes placed in different solute concentrations. You can see the x-axis is so this is the solute concentration running this way, and the y-axis is the change in mass. So if the isotonic point is where the there is no net movement of water, that means that the potato won't change mass. So that would be at this point right here. You see it right there. So if this is the point where it doesn't change mass, then what we could say is that roughly the concentration inside this potato is about 1 point, this is 1.5 by the way, 1.5%, that's 1%. It looks like it's about maybe 1.1 to maybe 1.2%. I don't know exactly if this is sodium chloride, what solute this, oh, it says salt, so it's probably sodium chloride. So what this graph says is that the potato has an equivalent of about 1.2% salt concentration. We can tell that because at that point, the potato doesn't gain or lose any mass. You can see that this is negative. So like these values over here show that the potato has lost mass, which means if it's lost mass, this must be hypertonic. The outside must be hypertonic because that would explain why the potato loses water. Whereas you can see up here, you got 2 points where the potato has actually gained mass. So that must mean that the potato is in a hypotonic environment. So like something like, for example, 1% would be hypotonic to the potato, which explains why the potato actually gains a little bit of mass. So just to wrap up, so osmosis and diffusion is a really important concept. There's something really interesting here about osmosis. So when a patient gets like a IV, you normally get an IV with, I don't know if you normally get it, but I've always seen whenever you get like a saline solution, IV, it's 0.9%. The reason why it's 0.9 is because that's actually the concentration of, the equivalent concentration of solute in your blood. So you don't want your blood to accumulate too much water, and you don't want your blood to lose too much water. You want to keep the cells, in that isotonic state. And we normally think about water like dehydration as a problem, but did you know that you actually die from drinking too much water. It's actually called water intoxication or water poisoning. Now, what happens in that case is because you're drinking so much water, the concentration of the salt drops. And the reason why is because concentration, which is C, is basically the amount of solute over the volume of your solvent. So the volume of your solvent increases, that means your concentration decreases. Now, why is that a problem? Well, because let's imagine that you have brain cells, okay? Imagine that this is blood vessel that is providing nutrients to those brain cells. Now, if the blood has become diluted because you've drank so much water, that means the concentration in the blood of the solute concentration is lowered, so it's hypotonic. And you can see there's a term hyponatremia. Hyponatremia just means that it has very, very low salt concentration. So we can just simply say the blood is hypotonic to the brain cells. And what does that mean? That means the water actually goes into the brain cells. Now, why is that a problem? Well, because brain swelling could lead to death. So apparently I looked it up at about 15 cases, I believe, of people who have died from drinking too much water. Who would have thought? Okay, so what we'll do next time we'll talk about ways the cell controls the movement of things going through the cell membrane, okay? Because diffusion and osmosis, the cell doesn't really have a lot of control over it. However, it can actually have some control over osmosis. Maybe we can talk about that next time. But that's it for now, okay? So we'll talk later.